Okay, so I'm going to be talking you through meiosis and how it is that we start with one regular cell and end up with all of our gametes. And remember, I say gametes, you say sex cells. Now, let's start with not a sex cell, we're going to start with a regular old cell right here. Here you can see we've got the nucleus, and of course, half of your DNA from mom, half of your DNA from dad. Remember, this is interphase, so it spends the majority of its time right here. Now, interphase is going to go on. We're probably familiar with a little bit of what we have seen before with mitosis. Now, this is going to be the start of what we call prophase I. Not just prophase, prophase I. And once you've seen probably this before, the chromosomes have thickened. You can start to see them. We've got centrioles there. We're going to see some spindle fibers in just a minute. You can see the nucleus is also starting to dissolve a little bit right there. So, you can see the chromosomes very visible now. I'm going to simplify this a little bit, and I want you to note what happened right there. I didn't screw up my drawing. I do that a lot, but I didn't right here. They're deliberately crossing over. That's going to come into play, and it'll be very important later on. More details on that in a little bit. But again, we're still in what's called prophase I. You can see the spindle fibers from the centrioles starting to extend. Going to continue that on so that you can see the nucleus is almost completely dissolved here. Remember, prophase, nucleus dissolves, so prophase I, very similar to that. What's actually gone on now, though, is when those chromosomes actually crossed over, they exchanged just a small amount of genetic material right there. Just that arm, if you will, of the chromatids switched over. And just that tiny difference is going to come into play and make a huge difference later on. Obviously, we're simplifying this, but that whole process right there is important. Now, again, all of this right here is still prophase I of meiosis. Now, after prophase I, things are going to kind of settle down into what we're very familiar with. This is going to be metaphase I, and if you remember, metaphase means that things are going to be lined up along the equator. So there are my centrioles and spindle fibers, just as we've seen before. After metaphase I is going to come anaphase I. That's not just a terrible circle, it's supposed to be an oval, because you can see the cell is starting to stretch. Spindle fibers have pulled them apart. They're no longer along the equator there. So that's anaphase I. After anaphase I, we're going to get to telophase I. You can see it's starting to pinch together. Don't ask how long it took me to get that. Nucleus temporarily reforming. Remember, that's what happens in telophase. It's kind of the exact opposite. And you can see right here that tiny switch is continuing to carry over. And now these are going to become separate cells. So each one is going to be a little bit different. There is telophase I. And of course, it's going to go through cytokinesis, and it's going to pinch apart into two cells. So here we are, end of telophase I. We're now going to start what's called meiosis II. So all that we've seen so far is meiosis I. This is meiosis II, very similar to what we know in mitosis. Prophase II, cells are going to continue to do their normal thing. This is kind of metaphase. They're lined up along the equator, if you will, and you're going to start to see those spindle fibers pull apart the sister chromatids. So here we are, metaphase II, lined up along the middle. Of course, after metaphase II is going to come anaphase II, when these cells are going to stretch again. Now you can see that after they've pulled apart, they broke apart at the centromere, and now they're going to opposite ways. So look at how I'm drawing this, because I'm being very careful about how I'm drawing this here. You'll notice that it got pulled apart. This one is getting a little bit of the chromosome from mom, while the rest of it is the chromosome from dad. And this right here, the smaller chromosome, is only the chromosome from mom. This one's the exact opposite. Still got our centrioles, still got our spindle fibers, anaphase II. So I got to scroll on down to make a little bit more room here to continue. So after anaphase II, of course, we're going to have telophase. I'm going to copy over my pinching cell because, you draw it right once. You might as well stick with it. So remember, in telophase, the nucleus reappears. So I'm going to start to see a nucleus form around these guys right there. And you'll notice each one of these is going to become a separate cell, and it's going to become a different separate cell. None of them are going to be identical. This is very different than what we've seen before with mitosis. So here I am at the end of telophase II. I'm going to have 4 cells, and each of them is going to be unique. You'll notice no 2 of them are the same, and it all has to do with that crossing over. And move that up and out of my way now. Now I'm going to show you here, because here it's a little bit different in men versus women. First one I'm going to show you here is going to be men. So I'm going to draw these 4 cells here, and in this case, they have tails. Those are not just crappy balloons, those are sperm. Now again, you can see those chromosomes are lined up there. Now this means that the overall process has 4 different sperm, all of them different from one another. That's what it's like in men. In women, it's going to be roughly the same idea, except you're going to have something a little bit different. You're going to have 3 really small cells, those are called polar bodies, and 1 massive cell. That is the egg that we all know and love, and we all came from. So you can see right here, the end result of this is 1 massive cell. It essentially sucks up all the rest of those. Those never get to be anything. And this big one here, but each of them genetically identical. Now we call these ones that kind of got shriveled up, they sacrificed themselves, we call them polar bodies and an egg, and then the other one became 4 different sperm. Now I want to zoom in on this part right here, this prophase I, because that is where everything is very important, that chromosomal crossing over. That right there is central to meiosis. It's what makes us all genetically different. In women, however, meiosis pauses here. And it pauses there for a while until you hit puberty. You, meaning a woman, hits puberty, and then once a month, one of those is to continue that whole long process through the process that we call menstruation. Now, I'm going to zoom in to focus on that crossing over during prophase I because that is central. So as we saw before, here is my cell. Remember, the nucleus was disappearing because this is prophase I of meiosis. Chromosomal crossing over. Very important right here. This is where the exchange takes place. So I've still got my centrioles. The spindle fibers are still coming out as we saw before, but the crossing over is happening right there. That is what makes all of those sperm and egg different. That's what I was talking about. Very, very important. Now, I want to show you just a continuation. So you got the crossover, and then you have, of course, the exchange of genetic material. This way it's just a little bit cleaner, a little bit easier for you to see what's going on. Crossing over, exchange sister chromatids. And remember, this is prophase I of meiosis, we're crossing over, and it's responsible for the difference in gametes. None of the sperm you will ever make, if you're a guy, will be identical. None of the eggs you will ever make will be identical. Very, very important. Thanks to crossing over in prophase I of meiosis. So, once again, you can see right here, there's where meiosis pauses and then continues on through this whole process. If it's a woman, then it becomes 3 polar bodies and an egg. If it's a man, it's going to become 4 sperm. But remember, unique. So men, of the 3,000 sperm that you are making per second, none of them are identical. Odds are pretty good you will never make identical sperm. Women, of the hundreds of thousands, if not millions of eggs you are actually already born with, none of them are identical. None of them are the same. This is why children look different than their siblings.